Alright, so far in our materials that we've covered thus far, we've identified some of the basic vocabulary in genotypes, phenotypes, homozygous, heterozygous, and so if we just kind of go through in a quick review of things that we've covered thus far, we've covered number one, we've identified incomplete dominance or not. If something is completely dominant, it's always going to win. So that's an example that we might have is stating that if we have in Mendel, green versus yellow. So a capital G is green, a lowercase g is yellow. We do a Punnett square. If we have a cross between a true breeding purebred green with a yellow plant, so I've got capital G, lowercase g, 100% of the F1 generation still should be the color of green, but their genotype is that. Phenotype, what do they physically look like, is green. Okay, that's an example of complete dominance. Now, when I want to move that along, we can also have an example of incomplete dominance. Okay, for number two, incomplete dominance is where the traits actually are going to mix. So this is our example that we've seen with flowers. If we have a red flower and a white flower, so we're going to have the genotype red here, white here, but when they mix in a Punnett square, we can get a brand new phenotype. So when a third phenotype shows up, that wasn't the original. That should give you the example that you get a mix, and that's an example of this. So if I have a Punnett square, big R crossed with WW, 100% of the offspring are going to be RW, or 100% pink. Okay, that's another example. So that's example number two. Example number three, what we've covered thus far, let's see if I can get my 3 to work, is codominance. In codominance, this is where we could also say multiple allele inheritance, where both alleles are going to show up. Now, our common example is blood typing, or A, B, O blood grouping. Now, I wrote O there just as lowercase to help us remember that O is actually a recessive blood type. So looking at our chances, if we had somebody who's homozygous dominant for A, but somebody that is heterozygous for B, example there, what are the chances of having a B baby? So if we go through, any chance of having a child with type B blood would be a 0% chance, because there's nobody that has the type of just B. I have 50% AB and 50% A blood typing. Those are the two that I actually get. Okay, so this is showing you an example of codominance. The last one that we talked about yesterday, we started taking a look at two traits, okay, which we call fourth, dihybrid. So we're looking at two traits. This is an example. If I have if I'm going to go with our G's, capital G is green, lowercase g is yellow, capital R is round, lowercase r is wrinkled. If I go through and have those as my key, I can take that point and let me small this a little bit better. I am going to cross one pea plant that is green, heterozygous for green, as well as heterozygous for round. I'm crossing it with another one that's heterozygous for both as well. Capital G, lowercase g, capital R, lowercase r. So when I got to figure out all my possible gamete combinations, I need to go through and I need to FOIL. So I take the first G with the first R. So I got G, R is my first one. I take the first G with the second R. Capital G, lowercase r. Then I got to do it again. I take the second G with the first R. So it's little g, big R. And then I take the second with the second. Little g, little r. Now I got to do the exact same thing over on this side. So when I do that, I take the big G with the big R, big G with the little r, and take the little g with the big R, and little g with the little r. So when I go through and I set up my Punnett square, now remember last time I gave you the actual equation when going through it, we always, in this example, going through, if I do the whole thing, keep your G's and your R's consistent. Always have your capital letters first. Going through, we can do the whole thing. I won't do it all, but I'll just kind of get you the idea. Ultimately, we are going to end in that one ratio, which I've told you before is always the ratio of 9, 3, 3, 1. But that is when both parents are heterozygous for both traits. That's going to be the ratio of the phenotype. So what do they look like? And remember what that ultimately says. 9 of them are going to be dominant for both. So 9 of them are going to be green and round. 3 of them are going to be green and wrinkled. 3 of them are going to be yellow and round, and 1 of them is going to be wrinkled and yellow. Okay, so that's what that's referring to in the dihybrid cross. The last one that we're going to talk about today is just in terms of review. We haven't spent a lot of time on it yet, is moving our way into what we call a sex-linked trait. Okay, sex-linked trait is going to be number 5. And what this is, sex-linked is referring to our 23rd chromosome. Our 23rd chromosome. So if we're going to go to a new page real quick. So our 23rd chromosome, remember on our karyotype, it's that big chart that we've seen. Okay, 1 through, I'm just going to say down here, is 23. We had 1 chromosome from mom, 1 chromosome from dad that made us diploid. Okay, we had that all the way along. And then we get down to the 23rd. Okay, all of the 1 through 22 are called autosomes. This one is called the sex chromosome. Okay, now this is the X, that is the Y. If I have 2 on the X, I have now a female. If I erase that, let's see here, we'll just do it old-fashioned way. Can I erase this out? We instead, if you can kind of imagine, I don't want to highlight. So scribble that out. If we had 1 X and 1 Y, we'd have a boy. So 1 of each. Now, what we're going to do is I'm going to give you an example of one of these types of traits in which this takes place. There is a trait called hemophilia. Hemophilia is the inability to clot your blood. Now, this trait is a sex-linked trait. Now, in a girl, remember, girls are X's. Boys are XY. Now, typically, boys are going to be affected by sex-linked traits more because they have 1 X. If this X is bad, they're stuck with it. However, if that X is bad for the female, they have a second one that they can correspond to, they can utilize. Now what's going to happen is when we go through and identify this trait, I'm just going to say big H is hemophilia, little h is normal. Now it really doesn't matter if it's dominant or recessive here, because if a guy has it, he's going to have it. If a girl doesn't, she can use her other X. So if I'm going to write out my genotypes over here, my possible combinations I have for a girl, I can be X big H, X big H. And what I have there is a female hemophiliac. I can have an X little h, X little h, which is a female normal. The last one I can have is I can have an X big H, X little h. Now remember we said again, if this one's bad, she doesn't have to use it. She can use this one. Ultimately, she's not affected by it, but she does carry it, so we call this a carrier. Now, carriers, please focus on this. Carriers are only utilized, we call this a carrier in terms of sex linked. If somebody's heterozygous, we don't call them a carrier. Okay? Even though they are heterozygous here, we're talking about the other chromosomes, the 23rd chromosome. All the 1 through 22, those autosomes, is when we can talk about heterozygous and keep it as it is. Now, if I'm looking at the genotypes of a male now, I need to go through and I do X, Y, X big H tells me I've got a male who's got hemophilia. I can have an X little h, Y, which tells me I got a female hemophilia. Or excuse me, female normal. I get a female who's normal. Okay? Female normal. So now what's going to happen here is I just kind of bring that over to the side. I'm going to say we're going to cross a male that is normal with a female carrier. Male normal. So X little h, Y with a female carrier. So I got an XX, got a female, and if she's a carrier, she's going to have one big H, one little h. So if I go through and do my Punnett square, what are the chances, now if I'm looking at this here, looking at this structure here, what are the chances of my boys suffering from this disease? So when looking at it, I'm looking at the boys only. Now, these are the girls, these are the boys. So the percent of the boys having it is 1 out of 2, or 50%. So here, that one there actually has hemophilia. What are the chances of the girls being affected by it? There is a 0% chance of any of the girls being affected by it. However, I have a 50% chance of having a carrier. Okay, so that's how this is kind of sets itself up, is looking at the X's and the Y's. Now, please make sure you spend some time. You go through it. We'll spend more time on the structure of it tomorrow. But I'm going to do one last one with you real quick. Okay, I'm going to say I have a female non with a male hemophiliac. Okay, going through that, is there any chance of having a male that has hemophilia? The answer to that should be no, because mom has to pass on the X, and both of her X's were normal. Okay, now if I have mom who's a carrier, which we had last time, okay, mom who's a carrier, she could pass on it to a son. So who usually gives sex-linked traits? Moms pass them on to sons, because dad had to pass on the Y. Dad passes on the Y, So moms are the ones that are the guilty ones to make you go bald, to give you hemophilia, to make you colorblind, so we can thank them.